Hello everyone and welcome to Biochem Sketch. I am an honors student in biochemistry and I will be presenting my own active study sessions as the raw, unedited, one-take videos. So just bear with me if I make a mistake, I will try and correct it. And if you want the PDFs of my notes, then the link will be in the description. So I'm just basically going to present to you questions that I've encountered and did not necessarily know how to approach the first time. So this was from a mock exam. So basically what they asked is, why would triheptanoin be preferred over trihexanoin to treat hypoglycemia? And you should explain it by showing how triheptanoin is metabolized. So basically, I'm just going to first explain this question to you, and then I'm just going to take you through some background undergraduate knowledge that may come in handy, that we all tend to forget during the years that we study. Okay, so first of all, trihexanoin is a 6-carbon molecule, and that therefore it is an even chain, while triheptanoin is a 7-carbon molecule, making it an odd chain. So during beta oxidation in the mitochondria, you must remember that the enzyme will cleave the carbon backbone every 2 carbons at a time. So for the trihexanoin, this will yield only acetyl-CoA molecules, whereas for the triheptanoin, the enzyme will cleave the first 2, and then when it gets to the third fragment, the third fragment, it contains 3 carbons, and it cannot cleave that carbon like over here. So this leads to the propanol-CoA forming. Okay, so you'll see now why I said on this side that since these acetyl-CoAs will all enter the TCA cycle, it will only spin the TCA cycle and immediately leave as carbon dioxide. And then therefore also no net oxaloacetate will be formed. So there's no oxaloacetate to spare for another process, and therefore it can't fix hypoglycemia, but you'll understand in a moment better why. Okay, so basically this propanol-CoA is now our molecule of interest. So we're going to take this propanol-CoA, and then the first step, we're going to do carboxylation. So what's going to happen is the propanol-CoA carboxylase enzyme will catalyze the conversion of propanol-CoA to dimethylmalonyl-CoA, which is a 4-carbon unit. So the CO2 makes it now a 4-carbon unit. This happens in the presence of biotin, the vitamin B7. And then the dimethylmalonyl-CoA will then be converted into L-methylmalonyl-CoA, which is also a 4-carbon unit. And this step is called epimerization. So the D isomer will be converted into the L isomer. And the enzyme at this step is dimethylmalonyl-CoA epimerase. Okay, so basically the only thing that happens here is the CH3 is now on the other side of the same carbon. So this is stereochemistry. Okay, and then we move from L-methylmalonyl-CoA to succinyl-CoA in the presence of the vitamin B12, and the enzyme is L-methylmalonyl-CoA mutase. So this is the mutase reaction. Now our resulting succinyl-CoA will enter the TCA cycle to replenish the succinyl-CoA over here, and then it will be converted into oxaloacetate during the various steps in the TCA cycle. And this oxaloacetate that is now more will be converted into glucose during the process of gluconeogenesis in the liver. And this then will elevate the blood sugar levels to counter hypoglycemia. So now you can see why this would not have worked, because the normal acetyl-CoA that would have entered the TCA cycle would just keep the TCA cycle spinning and yield cofactors like the FADH2 and the NADH2 that is necessary for the ETC to run and provide more ATP. But it would not have made extra oxaloacetate molecules. Okay, so basically now, what I first want to show you is just background knowledge of why somebody would have hypoglycemia. Let me just scroll there. Just see. Okay, so basically, if they have the pyruvate carboxylase enzyme, a defect in this enzyme, then they'll have trouble with hypoglycemia. So a healthy person will be able to convert pyruvate into oxaloacetate in the TCA cycle to refill it with new carbon skeletons. So remember, anaplerosis is refilling. But it will also help the start of gluconeogenesis in the liver to maintain blood sugar during fasting. So pyruvate will be converted into glucose. But now in an unhealthy person, or yes, in a patient that has a defect, this pyruvate can't be converted into oxaloacetate in the normal way because this enzyme is blocked. So now we need to follow the backdoor mechanism that I explained, with triheptanoin being converted into propanil-CoA, and then into succinyl-CoA, and then into oxaloacetate. So if pyruvate were converted into acetyl-CoA instead, then you would have gotten energy, but you would get no net oxaloacetate that could be converted into glucose. So this is very important for insight-based questions because they basically just want you to know that the triheptanoin will eventually form propanil-CoA. So this is your star of the show because it can lead to oxaloacetate producing more glucose. Okay, now I just want to show you another example. So this 4-methyl octanoate molecule. This is an even-numbered carbon, and this tripped me up first actually because it's a total of 9 carbons, and then you think to yourself, but this is not an even number. But the branch actually does not count. It's just about the straight chain when you name it. So this is actually an even number carbon, and it prevents the standard formation of acetyl-CoA at that position, at the branch position. So it'll also form propanil-CoA. It'll form 3 acetyl-CoAs and 1 propanil-CoA. I might explain this in the next video because it also came up in the test. So it is also anaplerotic because it forms the same propanil-CoA that I showed you here. That'll then be converted into oxaloacetate and then into glucose. But it has a little bit of a different mechanism, the oxidation mechanism. But it's not important for this video. Okay, so now let me just take you through the nomenclature. Okay, so a fat versus a fatty acid. This also I was like, I don't know. So a fat is a triglyceride, and the prefix will be tri and the suffix will end in in. So in our example, tri is the prefix and it ends in. So you know that this is a fat. So this is a storage molecule. 3 fatty acids are attached to a glycerol backbone, and a fatty acid's name will end in ic or ate, like heptanoic acid or octanoate. And the 8 is only signaling that it's in the salt form. So it is the individual string of carbons used for fuel. And then I just want to show you the other thing. Okay, decoding the chain. So the first part will tell you how many carbons there is. So hex will tell you that there's 6, so it's an even chain. And hept will tell you that it's 7. 8 is oct. Palmit is 16, also even. And ster tells you it is 18, so it's also even. And then just remember that I told you about the 4-methyl octanoate. So the moment that it's branched, the branch doesn't count for the even or uneven amount of carbons. It's just still an even chain, even though the total is 9. Then a short chain fatty acid is 2 to 5 carbons, medium chain 6 to 12, long chain 13 to 21, and then very long chain is more than 22 carbons. Okay, so this matters because in the for a first year student, an even chain fatty acid can provide energy, but for the postgrads and senior students, odd chain fatty acids like heptanoic acid provides energy, but it also has the ability to make new glucose to counter hypoglycemia. So that's why it's important to know this. And then substituted chains will also have other groups, like 6-aminoheptanoate has an amino or a nitrogen group at the 6th carbon. And then unsaturated means it has double bonds. So you must look for something like inuate instead of anuate, like 2-E-octanoate or 2-Z-octanoate. So this means the E and the Z just tells you the shape of the double bond, so it's similar to cis and trans. Okay, so I hope I covered most of this now. Let me just see. Okay, so this last one. So identifying the extras, branched and modified chains, you just look for methyl or dimethyl. So the example is 4-methyl-octanoate, which is an 8-carbon chain with a branch at the 4th carbon. So it's also useful for anaplerotic treatments. And I think this is mostly it for today's video. I just wanted to show you this entire thing because it's very useful during the very complex multiple choice questions during your honors year, and all of this becomes very important then. So yeah, I hope this video helped you, and if it did, please subscribe and like my channel, and you can get the PDF link of this notes also in the description box. And this will be only like for the price of a cup of coffee. So yeah, hope you enjoy, and good luck with the revision for exams.